Hi, welcome everybody, and thanks for being here for Technion Canada's first national online event. My name is Steve Bramson, and I'm a member of Technion Canada's Board of Directors. I'm also the proud father of a Technion Master's degree holder. Today we have a brilliant speaker, Josué Sznitman. Following José's presentation, he will be answering some questions, and you can pose your questions using the Q&A button that will present itself at the bottom of your Zoom screen, and you'll see that pop up when you mouse over it. Abigail Aronoff, our Montreal-based professional, will consolidate your questions and then moderate a conversation with Dr. Sznitman. Again, she will be pulling questions submitted via the Q&A button, not the chat button. The chat button is used for communications between participants. And at the end of the session, I will be announcing our next event speaker. So please stick around for that. Now I'd like to welcome Alyssa Greisman, Technion Canada's National Executive Director, to say a few words.
Thank you, Steve. This is Technion Canada's first national virtual event, and I'm thrilled to have you all here today to learn from one of our esteemed professors from the Technion Israel Institute of Technology, Dr. Josué Sznitman. Thank you to our board member, Steve Bramson, for being our host today. Thank you to the National Event Committee, Steve Bramson, Ronnie Kaplan, Steve Merling, Michael Schwartz, Erwin Tobin, Eran Gershon, Dean Lurie, Shauna Novak, Sam Spanglet, and Jason Bazerman. And thank you to the professional staff, my team, Abigail Aronoff, Judy Charney, and Jennifer Gallant for all of their hard work. This is the first in a series of virtual learning opportunities that Technion Canada plans to bring to you. Through these events, we will showcase Technion's world-class scientists and how they are working to advance education, critical research, and technologies that will change our world for the better. At Technion Canada, we are proud to raise support and awareness for the Technion, Israel's preeminent institution of science and technology, and a driving force for Israel's economic growth, national security, technical prowess, international presence. I invite you all to get involved with Technion Canada and help to support this remarkable institution. I will now ask Steve to please introduce our keynote speaker.
Great, thank you, Alyssa. It is now my great pleasure to introduce today's speaker, Dr. Josué Sznitman. Dr. Sznitman is an associate professor and researcher at Technion Israel Institute of Technology's Department of Biomedical Engineering. He is a graduate of MIT and the Swiss Federal Institute of Technology. In 2008, Sznitman joined the University of Pennsylvania as a postdoctoral fellow and moved to Princeton University as a lecturer and research associate. He joined the Department of Biomedical Engineering at the Technion in 2010 as a tenure-track assistant professor and was then promoted to associate professor with tenure in 2016. Sznitman currently serves as Associate Chair for Undergraduate Studies in the Department of Biomedical Engineering and is the director of the Norman Seidman Graduate Program for Nanoscience and Nanotechnology. As a researcher, he directs the Technion Biofluids Laboratory. Sznitman was awarded the Young Investigator Award in 2015 by the International Society of Aerosols in Medicine, and most recently, he received the 2018 Emerging Scientists Award for Drug Delivery to the Lungs by the Aerosol Society in the UK. We are thrilled to have Josué with us today. Just a reminder before we begin, any questions for Dr. Sznitman can be asked through the Q&A function at the bottom of your screen and will be addressed at the end of the formal presentation. So welcome, Josué, and thank you for being with us today.
Hi, everybody. Thank you so much for hosting me. It's an absolute pleasure. And if you hear some noise in the background, I have one of my boys playing Fortnite, but he promised he'd be quiet about it. He might pop in at some point. So I thought people would appreciate a little bit some context and although the topic is, let us say, rather heavy and loaded, nevertheless, I wanted to give everybody a sense of something that would be conveyed in layman's terms. So the first thing you need to understand is, as Steve mentioned, I'm not born nor raised in Israel. I came here at the age of a few days shy of 30 years old when the Technion hired me. I'm born in France and raised in the United States, and my folks moved to Switzerland when I was age 10 and basically have been there for the past 30 years. And this summer marks the 10th year since I made Aliyah to Israel. I actually have directly and indirectly strong ties with Canada. I have one brother and he's married to a Canadian. My brother studied at UBC. And for the anecdote, before I came to the Technion, I interviewed for faculty positions both in McGill and University of Toronto. But those are different times, and here I am. So you're wondering, how did this guy end up in the field of lungs? Maybe. I'm a mechanical engineer by trade. And quite frankly, nothing set me to go into this direction, because you see, my kid's dream was to design race cars. And that's practically the only reason I thought I should become a mechanical engineer, was because I should understand something about mechanical engineering to do race car design. And so how does race car design have anything to do with the lungs? Well, if you scratch a bit the surface, you'll understand that when you draw the shape of a car, although for me it was mostly about aesthetics and beauty, actually what it's doing, amongst others, is answering the laws of aerodynamics, which is being sleek in practice means resisting less to flows and air. So maybe now you're starting to get the gist of it, because really, if we're talking about physics of flows and airs, well, oh, there's Liam passing by. Then, oh, you want to say something? Hi. Yeah, you're playing Fortnite? Allez, va. So the lungs are what? They're an organ that basically revolves around inhaling and exhaling air. So they must obey somehow the laws of physics. And to make a long story short, I kind of came to realization that I was not going to be the next big Ferrari, but what I wanted to do was have an impact, a tangible impact on people. I was driven by science. I was driven by research. That's what I liked. And I had this love for the physics of flows. So I kind of took the physics of flows and twisted it to go and basically kindle with physiology, with medicine. And so I just jumped and pragmatically picked an area where I thought I'd have a small chance to make a little difference. And I was faced with basically a fork with two paths: either go into the field of cardiovascular flows, which is by far the better known. And there I thought, I'm nobody and I have no background, and there are very famous people there. I'm not going to make a difference. So I went for the other one, which I was curious about, but it was nevertheless a pragmatic decision. And here I am, many, many years later, basically 13 years or so since I finished my PhD, and I'm to some degree an expert, whatever that means, in lung physiology and the business of delivering drugs to the lungs. Okay, so I hope with that you get a picture of kind of the convoluted picture of my track record and why I'm in this business, which I absolutely love. And so where does COVID-19 fit in this? Okay, so one thing needs to be very well understood here is the angle in which my contribution has a potential. Mine, I speak in the name of my lab. In many ways, I'm just a PR machine of really the brains and the firepower and the heavy lifting that's going on behind in my group. But for those unfortunate people who suffer from COVID-19, what we call the severe patients, what happens is, as we know and we've read, like we've never read before, they end up potentially on ventilators via endotracheal intubation. And in the worst cases, they need to be actively anesthetized. In other words, they're not even conscious. What this means in practice is that their lungs are at the edge, if not, they're suffering from organ failure. One of the first metrics or ways of understanding this is that just the oxygenation level is gone really sour. Now, this condition of this lung failure, which has many, many implications, many, many symptoms that are not always the same between diseases, because in pneumonia, you can fall in this category. You can have a car crash and fall in this category. You can drown and fall in this category. But it's all under the umbrella of this syndrome called acute respiratory distress syndrome, which has many ways to get to it, but it shares similar characteristics. In the case of COVID-19, there are many of these characteristics that lead to this kind of whole lung organ failure, which is this kind of crazy immune response with immune cells rushing in. And one of the unfortunate events that happens there, and we don't understand everything, this is medicine, but nevertheless, the tissues get structurally damaged. And amongst other, you have fluid buildup, you have basically blockage of passing oxygen. And one other attribute is that the cells don't function. And one of the functions that they need to do is secrete the so-called surfactant. Now, surfactant, you've heard probably of this word, whether it's the lungs or in an industrial setting or when you use your dishwasher. But basically, human lung surfactant has many properties, among which one very tangible aspect of it is that it's like a soap, and it wants to basically help reduce the energy that requires to displacing a surface. I understand this sounds a little bit complicated when I say it, but we understand that when you put a drop of water on a table or on your shirt, if there's a stain without the soap, the water just wants to sit in a little bubble. When you add the soap, then it has the tendency to spread. So it relaxes the amount of energy. In the lungs, believe it or not, you really need this because you have a surface, a very big surface. In an adult, it's about 100 square meters. And it needs to have the surfactant so that it eases the energy that your muscles need to pull, okay? Without human lung surfactant, you simply cannot breathe. You don't have, your muscles cannot open your lungs. And this is one, it's not the only aspect, but it is one critical aspect in the syndrome ARDS, which I come back to the COVID-19. One second. Les garçons, s'il vous plaît, j'ai besoin de parler au calme parce que sinon je ne peux pas. Excusez-moi. Là, en plus, je suis devant l'audience canadienne. Donc, So, there's a lack of surfactant, which means you cannot breathe on your own, okay? Now, one crazy thing is that there's an analogous syndrome with the lack of surfactant, which exists in premature babies, in the newbies, in the neonates. And there, we've known how to tackle this for 30 odd years with near 100% success. So this is not COVID-19. It's not exactly ARDS, but it is called IRDS, Infant Respiratory Distress Syndrome. It shares several things, but the fact that the lungs were born too young, too soon, means that the cells are not mature enough to secrete the surfactant on their own, okay? So what have we come up with? Not me per se, but for the past 30, 40 years, we do this. We basically take animal-derived surfactant, which is extracted, and we inject it in the lungs of the newborn babies. Now, I'm not saying this is not an invasive maneuver, but it is certainly a life-saving maneuver, and it has near perfect yield, which is spectacular, which is also why you don't hear much about it unless your children experience that. Okay, so the idea is you deliver surfactant, relieve the energy needed to open and close the lungs, And by some magic, which again, in biology and medicine, we do not understand everything, but by exposing the cells to the surfactant, you also trigger their secretion. So it's like you jumpstart the engine. All right. So this strategy of doing a rescue therapy in the newborns is very successful, but it has always failed in adults. Okay, so this is our starting point. There are no therapies to treat ARDS. There's about 40% mortality rate, which is what you're hearing once you're on the ventilator in the news. Okay, so there's no therapy. What you do is you try to oxygenate with a machine and you hope for the best. This, in essence, is the status. If you fall in on ARDS, let me tell you, it's trouble. It's trouble because it's basically pseudo a coin flip. So we have not been able to scale up the delivery of surfactant, which is hypothesized to be a good strategy to try and rescue and heal the ARDS. Again, it is not healing COVID-19. The strategy here is if you become severe and you end up on ventilator, do we have a therapy to rescue your organ so that once you're back on track, the body can get back to taking care of COVID or the coronavirus or what we call SARS-CoV-2. So again, my contribution here is the rescue therapy to get people in a situation where their lungs can refunction enough that the body can start taking care of other things. Because once in ARDS and you don't have oxygenation, then you can imagine that the other organs are going to start to be in a dire situation because you may have multi-organ failure. So the idea was as follows. If potentially surfactant works in adults, which has not been proven, our hypothesis was perhaps it's not that the surfactant in itself doesn't work, it's just that it's not getting to the places where it needs to be. Now you have to understand, I'm not a particularly big guy, I'm about 1.73 meters, but you can basically visually see that if a newborn baby, his entire lungs is the size, the tip of my finger, because this is about a 2-kilogram person at best, anywhere between 500 grams, yes, and 2 kilograms. It is quite a distressing scene to see a neonate, but the lungs are the tip of my finger. So you see the tip of my finger against my chest, and you wonder, why is it that surfactant rescue therapy does not work in adults? And our hypothesis was simple. We're just not getting it to where it needs to be, because once you squirt endotracheally liquid, any of you who's not ever done any physics or fluid mechanics or flows, I think you can all grasp that liquids will always drain where gravity pulls. It's what our entire life tells us when we look at rivers and whatnot and your sink and so on and so forth. And so basically the idea is we need not to change necessarily the medication, or in other words, the therapy, which is the ARDS, which is a surfactant, but rather find a mechanism to deliver it appropriately in the lungs. And this is where it ties in with our work. I'm not a pharmacologist. I'm a biomedical engineer that tries to reconcile, if you have something you need to deliver, can you think of strategies to get it there? And so the idea was, if liquid is too heavy, then we need something that potentially is light. And so, not to my credit, although my name is on the patent, I guess maybe I fostered the discussion, but the inventor, my former PhD student, and now the lead researcher on the project, he thought, well, foam it, because basically foaming will take the same volume of liquid, but suddenly mix it up with a lot of air. And you can imagine that surfactant, by definition, will easily foam because it's a soap. And this is everything you guys know. When you wash your dishes and you put soap or you take a shower or whatnot, then in essence, you'll get soap immediately. And so here we are 3 years ago working again on ARDS, nothing to do with COVID, okay? ARDS is actually an orphan disease, okay? It only has in the United States maybe 200,000 cases per year. So it has nothing to do with cancers and so on. So it was always overshadowed. But there's one big but, it's that the costs related to hospitalization when you have ARDS are dramatic. And that has to do with the fact that you stay very, very, very long hospitalized in the ICU and you need a lot of care. So small population, giant, absolutely ginormous budget to take care of these patients. Okay. So you've got foam, and the idea is instead of squirting liquid, you're going to push foam into the lungs. And so there we are. We were funded by the European Commission, and then later on by the Ministry of Science and Technology of Israel in the early phase, and some private donors from the Technion were maybe chronologically the first who gave us a seed grant. And we put the patent, the provisional patent, in 2017. And in the first years, we tried to do this. And in January 2020, okay, so not even COVID, right? Not even COVID, we finished the first preclinical in vivo experiments in rats to show safety and efficacy of this delivery method. And we did it with success, which was amazing. And you ask, why the rats, right? Because in the scientific community, the starting point is you need to use an animal model that is established to show indeed first safety and efficacy of this treatment. In other words, we were able to show that in rats, which are kind of basically the size of neonates, we could deliver successfully as good, in fact, even slightly better than doing liquid instillation, in other words, squirting. So as we speak, what happened is that suddenly when COVID started hitting every country and ARDS was becoming, well, as you know, it being on a ventilator was suddenly starting to be big in the news, we got propulsed to push as fast as we could to move from now the rats to pig trials, because again, we can't just jump to humans just yet. What we need to show now is that in models of ARDS, so what you do is you induce a state similar of ARDS by removing all the liquid surfactant from the lungs of the animal. So you're basically doing a giant damage to the lungs. You induce this state, and then you administer the drug. In other words, the foamed surfactant. So as we're speaking, we're setting up the pig experiments in the Faculty of Medicine near the Rambam hospital in Haifa, shall I say, right across the trial for pig experiments. Now, what does it mean? It means that if we can show in this pilot study that, s'il vous plaît, les garçons, s'il vous plaît. If we can show in this pilot study that the pigs yield successful rescue, or we can rescue the pigs, we have a case to move to clinical trials in humans in the style of a compassionate treatment. Because for those who anyway are on a toss of a coin and there's nothing else than just oxygenation, then this might rescue their lungs so that we can get back to taking care of SARS-CoV-2. I think this wraps up the big picture of giving you kind of in a nutshell, what this liquid foam therapy is all about. In particular, in the framework of COVID-19, this is what we're after. We need, and we hope that within, give or take 3 months of experiments, because you need to do this scientifically in an accepted framework. In other words, you don't just, as you've heard in the press, various treatments that work, not all of them were done in a rigorous scientific manner with basically positive and negative controls. So we're doing it, we need to show that the liquid foam therapy against a negative control, in other words, just letting the animal unfortunately succumb from this ARDS, or against the state of the art, which would be squirting the liquid, we do better. So we're talking a trial of about 10 to 15 pigs that needs to be done over the next 3 months. I don't know what's going to happen. Let's be honest. I need to be, it's very important to keep our head on our shoulder. This is science. We do not know yet how it's going to pan out. Okay. We're very hopeful. What we have shown in ex vivo, in other words, we have excised the lungs of pigs and compared visually by staining the surfactant into the lungs, that in principle, the liquid foam therapy, its distribution across the lungs is many folds better than if you just squirt. So there's hope, but at the end of the day, we need to measure the oxygenation levels, the compliance of the lungs, and show rigorously that, we can make some magic happen. So I'm just as eager as you might be. But as a scientist, we need to be very honest. We don't know. It is what it is. But if it does work out to be, then quite frankly, the stakes are rather high. I think this really is the bulk of the story. I think it's best that we move on to the questions, and I'll be happy to take and try within the best of my capacity to answer whatever your questions might be.
Wonderful. Thank you so much. That was amazing and very accessible, even for me, who is not at all medical or scientific. And also on behalf of working parents, well done.
Definitely.
So I have a few questions here from the audience. The first one is, and I'm just going to read them as they're written. What are the preferred surfactants for this treatment? Any irritation concerns?
Okay, so it's a great question. Actually, there's one superb advantage of the therapy of delivering surfactant. We are a middleman in the sense that we are buying and using the same FDA-approved surfactant that has been used for generations, which is bovine-derived surfactant made by a few pharmaceutical companies, and foaming with our IP, so with our patented technology and the machine that distributes it, what is actually used safely and for generations now. So we're not reinventing the wheel. We just found a clever way to try and better deliver it.
Okay, thank you. Another question from the audience. Any plan to use this technology to deliver additional drugs into the lungs?
Absolutely. It just turns out that ARDS was the first embodiment of a therapy we could easily deliver because we could directly foam the surfactant. But actually, if you think on the future of the technique, we patented this along the lines of a generic drug delivery system. And what I mean by that is we use also bio-inert, in other words, they're not have any active ingredients, and it has a safety profile. So you can take this foam, take it in, and embed in the foam other medication. For instance, could be steroids, could be large particles that one cannot use via inhalation therapy. And one of the tracks, and again, this is borderline science fiction in the sense that we don't know yet, but regenerative therapies along the lines of delivering stem cells, which we don't know how to deliver via the lungs topically. What we do is we do it systemically. But the thing is, these are large particles and they're very large cells, excuse me, and they get easily trapped in the small capillaries of the lungs. So potentially, we have a very efficient way of delivering all sorts of biological matter and medication directly on the air side of the lungs. For various, it could be maybe one day for chronic obstructive pulmonary disease, idiopathic pulmonary fibrosis. Actually, the list is very lengthy because in many cases, such as COPD, the truth be told, we have at best palliative care, at best.
Interesting, thank you. So I'll just keep going. There's a bunch of questions here. Do you have any quantified model regarding the dosage of surfactant in IRDS?
Yes, absolutely, because those are already set by the clinical community. Now, I'll put a caveat to that. As many of you know, in medicine, there's a window of how much you do dosage. For instance, in the typical case of newborn neonates, we're talking about 1 to 2 milliliters of surfactant per kilogram. In practice, what it means for a full adult, we need to shoot about 200 milliliters of surfactant into the lungs in liquid. The volume is, of course, 4-fold bigger once you have air with it, but the basic dosage is on the order of a few milliliter, 1 to 2. We hope to use the least possible because it is very, very expensive. Surfactant is very expensive. And so the lesser surfactant we can use to show positive results will be the better avenue, even from a cost perspective.
Okay. We have a question about how oxygen inside the foam bubbles, so how does oxygen inside the foam bubbles interface with the lung surface?
That's a fantastic question. There's 2 aspects to it. First of all, you need to understand that as much as the foam, there's a preconception that the foam will drown you, that it's like this massive thing that will completely obstruct the lungs. It's not at all that. The lungs, when you breathe, are many liters. You have a baseline of about 3 liters. And when you breathe fully, you're already at 3.5, 4, 4.5. It depends how much inflation you need to do. But we're talking all in all, half a liter that's going in. So it's actually a very small volume compared to the size of the lung. And you're ventilating behind this foam that's passing through. And the foam itself is oxygen filled. So actually, there's no concept of potentially suffocating. There's none of that. That's just a discomforting thought because we're not used to this idea of inhaling a foam. You'd think you'd be choking, but it has nothing to do with that.
Okay, thank you. There's a question here about Technion working with other Israeli universities. Are you working in partnership with any other labs or universities?
So I'm working with hospitals in Israel, including the one in Carmel and down on the Faculty of Medicine. Beyond that, I work or I collaborate with various people in the field of lung research that have to do with the field of surfactant. But in essence, this liquid foam therapy, it's a wild idea. And it was, to our credit, in many ways, it's unheard of. It's unheard of because I'm extremely critical about the field of respiratory therapy because most of us in the field are. It's actually one of the fields that unfortunately has the least new therapies brought to market compared to all other fields.
Why do you think that is?
It's not so much why I think that is that somehow the pharmaceutical industries, for various reasons, it's a field where we've not invented many new drugs. It's been mostly relying on repurposing drugs and trying to find a reusage of it. And it has a super high failure rate in going from preclinical to human trials that are successful. This is well known over the past 50 years. So I guess in a way it has room for really disrupting. But the idea of putting inhaling a foam was just so out of the box that nobody had ever, it's an idea you just hadn't seen. So that's to the credit of Jan. I can't take that credit for thinking that wide. I may take just the credit for having hired the right people. I'll take that credit.
It's an important step.
It is.
We have a question relating to sort of how out of the box this therapy is, and what's the estimated time to commercial release for this product?
Well, as we speak, we established the company. It's called Neshima Medical. Neshima, for those Hebrew speakers, means breathing, breath. So it happens to sound Japanese, which was what Jan wanted. So we've got a Hebrew name company that sounds Japanese. And Nishima Medical was established, and we're raising the seed round in parallel with what's going on in my lab. And the way it works is that there's a license agreement between the Technion and Nishima, where Jan, again, this guy, is going to be the acting CEO. And the license agreement, basically Nishima is using a license to use liquid foam therapy. That's the working horse of the company.
Okay, thank you. And I guess in line with that is the timeline to human trials when we move from.
Maybe if everything were to work, the end of the year, 2020, that's a very optimistic view. But
That seems fast from what I've heard. Is that because you're being fast-tracked because of COVID?
Yeah.
Yeah.
If by some miracle the pig trials come out positive, even in the pilot, so it's a small pig trial, okay? It's not even a full scale. There's a grounds to have a fast track for compassionate treatment, because in the eyes of it, well, with no other alternatives on the table, so it's that or nothing.
Okay. I have, we just got a slew of questions. I'm glad we have some more time. I will read this question verbatim because I'm not sure even I understand it. In addition to reducing surface tension, surfactant has been shown in some studies to enhance lung immune and defense responses. Will you examine this possibility in your pig model?
Okay, it's very true. Actually, this surfactant is so magical that all the artificial trials of trying to make an artificial surfactant have never been ever as closely successful as using bovine-derived directly animal model, because there's so many small molecules that are, maybe 1 part per million, that have all these small intricacies that we even if we think we know what are the basic ingredients, the different proteins, etc., the phospholipids, it doesn't have all the magical ingredients to make it complete, among what one of your attendees has said, which is the immune functions, which are extremely relevant, which we understand partly. In the first level pig trial, we are monitoring basically the ground level metrics to rescue. So it's not on the immune side as much as it is getting the oxygenation levels up, the lung compliance back to normal, and getting to a point where the person can breathe on their own. Of course, the hope is that's level 1, and then you have the different layers on top, which is, well, you're giving now immune functions. Potentially, you can trigger a stronger immune response that potentially can start working back on SARS-CoV-2. But it's a black box. We don't know. We don't know.
Okay. Do you have time for a few more questions? One or
Sure.
two? Okay. So we have one that's sort of a two-parter, and you talked about how the delivery feels a bit strange and new to everybody. So a bit more information is asked about how is the foam delivered to the body, and also, is this a medication that is prepared or mixed before it's delivered, or is it pre-packaged and you can just sort of ship it around?
These are excellent questions. They really are, because it really shows that the people are visualizing the way it's going to be given. And the answers are very simple. For the first part, the way it works is, again, when you're talking about severe patients, they are intubated endotrachally, and they are anesthetized. So what you do is, in the intubation tube, you connect the machine. This is a medical device that pushes, with the ventilator, that pushes the foam into the tract and the trachea, into this endotracheal tube. So it's just following the line, and behind it, you're ventilating with the normal ventilation machine. So it's synced with in that way. 2, how is it done in terms of the foaming? So it's in many ways the off-the-shelf surfactant, And the device, this proprietary device, foams it on the spot right before the administration for the patient. So it's like vial-based use. You load it, you foam it, you give it. It's not sitting around in foam. It is foam for the purpose because it's very important that they be delivered in foam. And then once in the lungs, bubbles like on beer and just like in the sink will resettle back into liquid form.
So it's an immediate delivery, right?
It is an immediate preparation and delivery.
Okay. And I have another question here is, do you know what the estimated time is to be certified by the FDA? Like, what is that?
Wait, I don't know. It's a little bit off of my comfort zone because Jan is really the biz guy at this point. But it's called, the track we're hoping for is probably in your audience, they know better. It's like a 505(b)(2) or something. In essence, we're not making a new drug. We're just administering it differently. So this is not a 10-year track. This is on the order, I think, of 2 to 3, under 5, something like that. Again, I'm a little bit off my comfort zone, but yes, that's definitely part of the company's to-do list. That's exactly what we're going for. Because we're at an orphan disease and we're not inventing, we're using an off-the-shelf medication that is already approved for God knows how long.
Okay. I have another question here that's less about the medical aspect, more about the university itself and its commitment to taking new research from the lab to the market. Are you comfortable talking a little bit about the university's reputation for helping that happen, or if you've...
I can speak in my own experience because this is what we've been working on. It's not the first time, but certainly I think Nishima is by far of my personal career, potentially the most successful endeavor and entity that goes from what you call from lab, that has a translational aspect. Yes, by all means, the Technion is extremely focused on succeeding in going from IP to startups. With various options on the table. It could be through an incubator. It could be directly licensed to another company. It could be through a startup. In our case, basically the Technion, and in particular, in this past few months, I have much to thank the Vice President, Kobi Rubinstein, for helping accelerate this translational this translational aspect, which is from the IP to the company, and the team at what is called T3, which is the Technology Transfer Desk, this is their business. That's what they're about. This is what they look for on campus in pushing technologies into commercialization.
Yeah, because a lot of what our Canadian community appreciates is the entrepreneurial spirit of Israelis, and in particular the university. So it's nice to hear that you're being supported. I have 2 more questions here. I know that it's getting on to 1 o'clock, but there's a question of what is the foam made of?
So again, the foam in the case for ARDS, it's surfactant.
That's all it is.
We just have basically proprietary technology to foam it the way we want it to be foamed. But it's literally, it's like the soap, and you would put your finger and stir and put a bit, and it becomes a bubbles. That's the idea. In other drugs, then what you use is a bioinert. It's actually one of the ingredients of surfactant, which is easily foamable, of human surfactant or natural surfactant, and then you would load inside that foam the active ingredients or the drugs of interest. But that's basically what it is. In this specific case of tonight's discussion, we foamed surfactant as is.
Okay, so that sort of almost exhausts the questions from our group. I think that based on their questions, you were very, very interesting and important conversation. Thank you for your time. I'm going to let Steve wrap up the conversation. But again, thank you so much, Professor.
And thank you very much for having me. An absolute pleasure.
Great. Wow, thank you, Abigail. And Dr. Josué Sznitman, Really, it's amazing. I'm inspired by how something that's in front of us, like you say, foam that we see in the shower or the bath, can then the inspiration of seeing how that could be applied for our benefit is it's just incredible. So on behalf of myself and all the participants, I want to thank you for taking the time to speak with us today and for providing insight into your groundbreaking research. The work that you and your colleagues are doing in support of COVID research is crucially important, and we are grateful to you and so many others at the Technion who are working to improve and save lives. So in that vein, we are excited to announce that our next webinar will be featuring Didi Mary, PhD. Didi is an associate professor at the Faculty of Biology at the Technion and is a member of Technion's Integrated Cancer Center. Dr. Mary is focused on investigating the therapeutic potential of phytocannabinoids. He operates the Cannabis Database Project, and his lab is currently involved in 8 clinical trials covering diverse aspects of cannabis treatments, such as colon disease, pain prevention, cancer treatment, and epilepsy. The event is, and mark this down, Tuesday, July 21st at 12 p.m. Eastern. Keep an eye open for your invitation. You won't want to miss this. And to help improve and shape such future events, please take a few moments to complete the brief survey that we will be sending you. You'll be getting those by email in the next few days. And I want to thank you, the participants, for attending today. And I encourage you all to share with others, from a distance, of course, what you have learned. Oh, wait a moment. There's one more thing here. A surprise. One of you has been selected a draw winner. And the winner today is... It's Helena Halpern. Helena, congratulations. You've won the coveted Technion prize pack. You will be contacted directly for delivery information, and well, thank you for participating. And that pretty much wraps it up. We look forward to seeing you all at our next webinar, and thank you again for participating. Goodbye from the Lisa Steinberg Wind Tunnel Control Room at Technion University. Well, albeit virtually. I'm actually in Montreal. Goodbye, everybody. Thank you.